So, hello students. Today we are going to start with monohybrid cross. That is, we are going to cross two plants with one character, and that is called as monohybrid cross. So let's start. When only one character is considered while crossing two plants. So you can understand here, you can see here, we are going to cross two organisms or two plants, and we are going to consider only one character. Such a cross is called as monohybrid cross. Here Mendel has considered height character. So Mendel has considered height character for monohybrid cross. He took pure tall plant. Pure tall plant has the genes capital T and capital T. And pure dwarf plant the genes are shown by small t and small t as parents. So remember, here we are going to take two plants and two pure plants. One is pure tall, one is pure dwarf, where capital T is dominant. Always remember, I have told you in the last class, capital letter is used for dominant character. So capital T is dominant and small t is recessive. When he crossed these two plants in the first generation, that is F1, he got all tall plants. We are going to see these all with the help of all the crossing and all by using the symbols. But first we will see the notes. When he crossed pure tall plants and pure dwarf plant, whatever F1 generation he got, in that F1 generation, all the plants were tall. Now, to check where the tall plants in the F1 generation exactly the same as the tall plant of the parent, means here we have taken pure tall plant as a parent, that is capital T and capital T. But whatever tall plant we have got in F1 generation, is it same as the parent? That we have to check. So now to check where the tall plant in F1 generation exactly the same as the tall plant as the parent generation, he self-pollinated F1 generation plant. So now what he did, just to check whatever tall plants he has got in F1 generation, are they same as parent? What he did, he took the plants from F1 generation and self-pollinated them to get F2 generation. So now we will start with first F1 generation. So Mendel has took pure tall plant. So I will write it here, pure tall plant, and he took pure dwarf plant. So that also I'll write it here, pure dwarf plant. So he took these two plants as a parent. So pure tall plants are shown by capital T and capital T. So write down, and dwarf plants are shown by small t, small t. So you can see here, small t means pure, capital T, capital means pure. So now take gene as capital T because both are same, capital T, capital C, we can write only one T. Here also both are same, small t, so we can write it as one small t. When you cross these two, and this pollination is called as cross pollination. This is called cross pollination. Why we will call as cross pollination? Because two plants are different. We are not doing the pollination in the same plant. We are doing the pollination in the two different plants. One is pure tall, one is pure dwarf. And that's why it is called cross pollination. So when we cross these two, we will get capital T and small t. So if you focus here, capital T and small t, both the genes are present in our F1 generation. So write down here F1 generation. So if you see the genes, capital T is dominant, small t is recessive. So which is expressive? Expressive is dominant one. And therefore, whatever plants we are getting in F1 generation, all will be tall. All will be tall plant. But this is not pure. This is not pure. It is impure, we can say, or we can say heterozygous. Homo means same, capital T, capital T. Small t, small t. These are called homozygous. Homozygous genes. And because this is capital T and small t, here this we can say heterozygous. gene. So this is homozygous, this is heterozygous. Now see, in F1 generation, we got all tall plants, but all are heterozygous. Now what we are going to do? We have to check this plant is also tall, parent is also tall. See here, parent is also tall, and F1 generation plant is also tall. So we have to check, is this tall plant which we have got in F1 generation, is it same as parent? And for this, we have to go with F2 generation. So now we will start with F2 generation. So see here, we are going to write it F2 generation. So I'll write it F2 generation. And for F2 generation, we are going to take the plant which we have got in F1 generation. So I'll write it here, capital T, small t. This is also capital T, small t. So we are going to do the pollination in the same plant. And that's why it is self-pollination. So we will write the genes. So see here, we will get two different now because one is capital T and one is small t. So the possibility is capital T and small t. Even here also we will write capital T and small t. Remember, these plants are tall. This is again a tall. Now we have to cross this, and whatever going to cross, this is called as self-pollination, because here the plant is same. Self-pollination, remember this is self-pollination. Now this we can cross with the help of checkerboard, or we can cross it directly also. First I'll show you how it is crossed directly. So first this T crossed with this T. Then this T is crossed with this small t. So what we will get? Here we will get capital T, and here we will get capital T and small t. Now see the another T. So this small t we will cross with this capital T. So we will get capital T and small t. Again this small t we will cross with this small t. So we will get small t and small t. So like this also we can cross, but it will be little bit difficult to understand. And that's why it is easy to use checkerboard. With the help of checkerboard, it will be very simple to understand. So we will see with checkerboard now. Now with the help of checkerboard, first write down the gene. One is capital T, one is small t. Then this side also write down capital T and small t. So these are the genes of two plants. So now cross it. So when we cross capital T and small t, when you cross these two, when these two genes will get mixed, here what we are going to get? Capital T and capital T. So write down capital T and capital T. Now what you are going to do? This T will combine with this T. So what you are going to get? Capital T and small t. So write down here capital T and small t. Now what you will do? This small t and this capital T. So write down capital T and small t. And last remaining is small t, small t. So write down small t and small t. Now if you focus, we are getting the same progeny as we have got here. So see this capital T, capital T. Here also it is capital T, capital T. Capital T, small t, capital T, small t, capital T, small t, capital T, small t. Small t and small t. So both are giving same result, but this is with the help of checkerboard, which will be very easy and simple and neat to understand compared to the earlier method. So this is called as F2 generation. Now we will write phenotypic ratio and genotypic ratio of this F2 generation. First we will see the notes of this. Now if you see the notes, till here F1 generation we have finished. Now see, the progeny of second generation, that is F2, are not all tall. So this is called as progeny of F2 generation, or this is called as progeny of F2 generation. If you focus these all are not tall. 1/4 of them are short. Now you can focus. See this one, capital T, both are dominant. So plant is tall. See second one, capital T, small t. This is also capital T, small t. But here there is one T capital that is dominant. Therefore, these both the plant will be tall. So these all three will be tall. But what about the fourth one? this has small t means it is a short plant or a dwarf plant. So out of these all four, one we are getting short and three we are getting tall, and therefore 1/4 of them are short. This indicates that both the tallness and shortness traits are inherited in the F1 plants. So if you focus here again for F1 generation, here it was pure tall, here it was pure dwarf. But when we got the F1 generation, what we said? We said all the plants are tall. The meaning is, here it is not that the short gene will not transfer from the parent. When we crossed these plants for F2 generation, here we got one of the plants short. Means the traits which are there in the parents are inherited to its next generation. Once I said all the plants of F1 generation are tall, means we should not think that only tall character has been transferred. When I cross this F1 generation plant for F2 generation, I got one plant as short, means tall as well as short. Both the traits of parents are transferring to their next generation. This is proved with the help of this F2 generation. So next, this led Mendel to propose that two copies of genes controlling traits are present in sexually reproducing organism. So here it is proved that two copies of gene, that is one is dominant and one is recessive, both are controlling the character where in sexually reproducing organism. These two may be identical or may be different. May be identical or it may be different. Then depending on the percentage. So what is the percentage here? If you see the phenotypic ratio, how they look physically here 1 is a dwarf but 3 are tall. So therefore 1:3. 1 is dwarf, 3 are tall. What is genotypic ratio? What is the genes? If you see capital T, both are same, it is 1. Capital T, small t, small t, these are 2. And this is small t is 1. Therefore, genes, genotypically if you see, it is 1:2:1. And phenotypically if you see, it is 1:3. So this is about monohybrid cross. I hope you understood. Practice this table, it will be very easy. Now we are going to start with dihybrid cross.